Welcome to section 11.2. So we've previously discussed what we'd call Mendelian inheritance. So this is when it's pretty simple, where it tends to be we look at single genes that cause a single phenotype and there's two different types of alleles. So we have height, and height is controlled solely by one gene for which there's two options, tall or short. Now this does work for many particular genes, but there's also going to be some genes that are non-Mendelian. So there's going to be some guys that don't behave in the simplest Mendelian way that we've talked about. And we call that simple Mendelian way complete dominance. So if there's a dominant present, it overshadows the recessive completely. To be recessive, you have to be homozygous recessive. But there's also going to be some other ways that get a bit more interesting. So two of these ways, the kind of the simpler ones, if you will, are incomplete dominance and codominance. Now, these are two that people screw up all the time. They know that, it's going to be one of these two, but they have difficulty figuring out which one. So let me try to go through and explain each. So incomplete dominance, this idea here is going to be one that's essentially blending. So you got to treat stuff a lot like you treat paint, where if I mix a black paint and a white paint, I get gray. So if you were to take a white allele and a black allele, you would expect to get a phenotype that's in between, gray. Now we can see this illustrated over here where if you are homozygous dominant, big R, it's a red flower. If you're homozygous recessive, little r, it's a white flower. But if it's a heterozygote, it's going to be pink. This is important and useful because it allows us to use phenotype to distinguish genotype. Prior to this, if something was red and red was dominant, I would have no way of knowing whether it's big R or big R because these were effectively the same. This is why our genotypic ratio was not always the same as our phenotypic. If you remember back to Mendel when he crossed the two heterozygotes just like this, he would have gotten 3 that were red, 1 that was white. But genotypically it would have been 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive, 1 to 2 to 1. So there was a difference there. But with this one, we can eyeball in most cases if it's a visible trait. We can eyeball it and say if they're pink, they must be heterozygotes. Phenotypically, this would also be a 1 to 2 to 1 ratio. So this is kind of a cool thing that we can do. Now the tricky part is there's another guy called codominance. Now codominance is going to be a trait where they both get expressed. So it's very similar to this, where we can have red if you're homozygous dominant, we can have white if you're homozygous recessive, but if you're a heterozygote, what we essentially get is a mix where both are expressed, so you get more of like a weird pattern where it's white and red. It's not going to be pink though. It's going to have red and white, but it'll be like splotched. So certain parts of the plant will be one type of the color and certain parts will be the other. The other way I explain this is if it's incomplete dominance, neither one gets full control. Neither one ultimately gets fully expressed. And so in that case, you get this middle ground. So tall and short make medium. If it's codominance, they both get fully expressed. So this would be like tall and short, and you have one long leg and one short leg. So I guess you're just crooked. But both are fully expressed. They're just fully expressed in part of the organism. Versus incomplete dominance, they're neither fully expressed throughout the entire organism. You get this middle ground where all of the organism is pink, all, or at least the flower, is pink, and so it all is that middle ground. So you've got to make sure you get comfortable with this because that's one of the most common mistakes is people will go, oh yeah, I can tell, that there's going to be these heterozygotes, but if it's pink like this, they've got to think, oh, it's incomplete. And oftentimes they'll just go with codominance. When you see something that's got this kind of more splotched pattern where there's red and white, not a mix of red and white, not pink, but red and white both present, at that point you've got to say, oh wait, this is going to be codominance. Now for an example of codominance in real life, we've got a particular disorder that's typically called sickle cell anemia or sickle cell disease. And this is when your red blood cells get this weird sickle shape because protein is changed in your hemoglobin. It's the stuff that ultimately helps you hold oxygen. It's one of the things in your red blood cells. And so once this changes, it makes it get this weird crimped sickle-looking shape. And that allows for it to clump up together. So the blood cells don't tend to live as long, so you tend to be low in red blood cells. That's the anemia part, is low red blood cell count. And you also tend to get these blockages that can form. And if they're not watched and treated, these blockages can lead to early death. So that's why this used to be a very bad trait to have sickle cell anemia, or sickle cell disease, if you will, where you were essentially homozygous for the sickle trait. And so if you were homozygous recessive for that sickle trait, that was the worst. Even nowadays, it causes pain, it can get you hospitalized, it's not a great trait to have, but you can stay on top of it so it's not as bad as it used to be. Now, in between this, you've got the heterozygote. Now, sometimes with codominance and with incomplete dominance, you'll still see this upper and lowercase idea, even though it's not really as meaningful anymore. So sometimes, too, they'll do something and say, like, all right, it's blood cells, so let's just use capital B, and we're going to use a superscript here to represent which version of it. That way we realize they're on equal footing. So you might say where you have, like, B with, like, an N here for normal blood cells, and a B where it's an S to show it's the sickle cell trait. And so that way we realize that you're not supposed to weight one more than the other, that they're both equally going to be there. So if you're a heterozygote, what we get is you get normal blood cells, and you also get sickle cells. So you have a mixture of the two. And so that's going to be essentially like here you can see some normal ones, you can see some that are affected. Now this actually is the best condition possible because you don't actually show symptoms of sickle cell because there's not enough of the sickle cells to really run amok, but you also have a resistance to malaria. And so because malaria has killed a lot of people throughout history, it's a really good thing to not have to worry about getting it, or at least have your chances drastically reduced that you'll get it. And so the heterozygote is the best trait to have if you live in an area with malaria, which is why we do tend to find this allele in certain parts of Latin America, parts of Africa, where this is a real fear, a real problem that you get and possibly die from malaria. And so these individuals that are within those cultures have been selected where individuals that are heterozygous have lived and reproduced better than those that are homozygous normal or homozygous for sickle. Now, if you live in an area without malaria, it's not worth the risk, so you don't tend to see this as much. But in those areas where it's present, there's a very valid reason to try to be a heterozygote. There's a valid reason to want to be this heterozygote for the condition because you're healthier. Now, if you are homozygous normal, which I'm going to go with my convention here for each just to get you guys used to it. So if we've got where you're normal, yes, your blood cells function wonderful and they do their job, but ultimately, so in this case, we've got effectively no malaria, and in this case, we have malaria. And that's going to be a major problem, as we said, for places that actually have the mosquitoes that carry the malaria protist, which then can get into your body and cause massive problems. So this is one of our examples of codominance. Sometimes they'll list sickle cell disease as being a recessive trait. They do that because you're only impacted if you happen to have two of the sickle cell alleles. So it does function very similar to a normal recessive trait, but it's not actually the case because when you look at the blood cells, you'll see a heterozygote does not have all normal blood cells, which is what we should see if it's completely dominant. Instead, what we see is a mix. They're all one or all the other. They're not half sickled. You know what I mean? It's not like each blood cell is just a little bit bent and a little bit skinnier and dies a little bit earlier. You're either a perfectly normal blood cell or you're a completely affected sickle blood cell. That's why we consider this codominance, not complete dominance, not incomplete dominance.